problem that many dairy producers face is the death loss of heifer calves. Death loss of pre-weaned dairy heifers costs U.S. dairy producers up to $100 million every year. Even when the proper management practices are in place, there's still a difference in survival rates among pre-weaned calves. This suggests there are other maternal and dietary influences that affect calf survival. Some of these other sources could be the maternal microbiome, the colostrum microbiome, and the colostrum components. We propose that these factors play an integral role in calf gut health and contribute to preventing calf mortality and morbidity. So what is the microbiome? The microbiome is a community of microorganisms that inhabit a particular environment, such as a human or a calf. For every cell in the body, there are roughly 10 more microorganisms that are essential to a healthy life, which make up the microbiome. The newborn calf's vaginal, fecal, and colostral microbiomes are the primary sources of the newborn calf's microbiome. Right now, there's very limited information on the long-term health effects of these microbiota on the health and performance of dairy calves. This is what we are investigating further in this study. The goal of this research is to identify the optimal bacterial phyla that impact long-term calf performance. Our hypothesis is that calf gut health is affected by the maternal microbiome with important long-term impacts on calf health, survival, and performance. We are aiming to identify the relationships among cow microbiome, and calf health, as well as determine relationships of colostrum composition and calf microbiome with calf health. To accomplish this, an experiment is currently being conducted at the Virginia Tech Dairy Center. This experiment includes 12 multiparous Holstein cows and their bull or heifer calves. These 12 calves are individually housed for 60 days after birth. Oral, rectal, and vaginal swabs were collected from the cows at calving. Colostrum was also collected at calving, and calves were fed 1 gallon of their dam's colostrum within an hour after birth. Swab samples were collected from the calves at 12 and 24 hours after birth and on days 7, 21, 42, and 60 days of age. These samples will later undergo RNA extraction for metatranscriptomic analysis to determine microbiome composition. This study is currently still underway, therefore the results will be coming later this year. So far, we have extremely healthy calves. There have been no instances of illness in any of the 12 heifer or bull calves. The average daily gain of the calves so far has been 1.49 pounds per day. All of the cows in the study gave, on average, 1.8 gallons of high-quality colostrum. Quality measurements of the colostrum averaged 26 bricks across the 12 cows. Once metatranscriptomic data is available, we expect to see that dominant phyla in the microbe samples will differ between sites but will be similar across cows within a site. We also expect that the cow microbiome will be similar to the calf microbiome. It is expected that the cow microbiome will influence the calf microbiome and, subsequently, calf health. The results of the study allow the optimal bacterial phyla for calf health and performance to be identified. This could possibly lead to the development of calf inoculation protocols and tools to provide pre-weaned dairy heifers with bacterial communities to support optimal health and performance.